Okay, everybody hearing me? Great. So thank you so much for joining me today, and thank you to the RCC for this wonderful opportunity to be in Munich and write Choral Whispers, or try to finally finish it. And thank you to my lovely family who has been dragged here and is sitting here with us today. So thank you for coming. I would like to talk today about one chapter from the book Choral Whispers. Some of you have already kind of read the last chapter of the book. This is going to be the first chapter of the book, chapter 1. And the book is tentatively titled, as Elena said, Choral Whispers, Scientists on the Brink. I welcome any comments about the title, which is contentious. So let me begin. In June 2016, I attended the International Coral Reef Symposium in Hawaii. That's not me. At this meeting, about 3,000 coral scientists, managers, and policymakers from 70 countries convened, as they do every 4 years, to discuss the present and future of tropical coral reefs. The meeting was much larger than ever before and quite overwhelming. Waves of coral people from around the globe and dozens of simultaneous sessions made it a frenzied affair. The only relatively quiet moment I had over this week-long conference happened, how else, in the restroom. You didn't mention my restroom work. We can insert a little a chip to my work on toilets here. Okay, there I had the first quiet moment to reflect on how unsettled I felt. What was it about this conference, I wondered, that got me feeling so disoriented? The answer came in a flash, and even had a catchy title: Biopolarity. What I experienced at the International Coral Reef meeting was the oscillation between the extremes of hope and despair. And since scientists are in the business of keeping corals alive, the term biopolarity seemed to be more adequate than bipolarity. I should admit to my own oscillation between the extremes of hope and despair. I grew up, as Elena said, in Israel and spent the summers of my teenage years scuba diving in the Red Sea in Sinai, Egypt. This is me. But life has taken me away from the ocean for 20 years, and so much has happened to coral reefs since then. I didn't realize the extent of the change until my visit to a coral reef in Culebra, a small island off the main shore of Puerto Rico, in January of 2015. I was there with my then 7-year-old daughter, Ariel, so you'll see how many years I've been doing this by how old she is now, to conduct research for my book, Wildlife. This is the cover for Wildlife, which focused on the relationship between captive and wild sites of nature management. Coral nurseries provided an interesting example, and I was planning to dedicate a few pages to restoration efforts in the ocean. So here we are, Ariel and I, exploring the elkhorn corals. Well, not right here, but you'll see in a second. Here are the elkhorn corals, and not such a great pose there, but me trying to explore a turtle in Culebra. Okay, you see how fieldwork can be very demanding. Yes. So the Puerto Rican coral scientist Edwin Hernando Delgado proudly showed me around his underwater coral nursery. He especially emphasized his efforts in transplanting Acropora species from the nursery into the devastated reefs. So here he is on the left doing work on his nursery. And here a close-up of the Acropora cervicornis. For most of the dive, a voice in my head kept repeating, "But where are the corals? Where are the corals?" I had it. Where are the corals? Okay. I was used to a much more diverse and colorful display. This sudden and direct realization that all is not well underwater prompted my decision to dedicate more time to learn about corals. The obvious choice for me was to interview and observe people who seemed to know a lot about these creatures, coral scientists. It helped that coral scientists are a tight-knit community, Edwin Hernando Delgado from Culebra was able to put me in touch with Australian scientists when I visited the Great Barrier Reef in May 2015. And here again, you can see how difficult the work of an anthropologist studying corals is. So here we are in the Great Barrier Reef. And this is in the green zone because this is kind of how GBRMPA, or the Great Barrier Reef Marine Park Administration, manages coral reefs through a lot of protection, kind of fortress conservation, what is called. I thought I'd show, so here's me taking a photo from the boat of the snorkelers. And I wanted to give you a sense of how the underwater sounds like or looks like. Yeah, video is not my strongest, but I'm getting better. All right, so Edwin also suggested that I go to the Red Sea, so I came back to the Red Sea and interviewed a few people there. Unfortunately, the sites where I scuba dived in the Northern Great Barrier Reef and in the Red Sea have suffered a lot since I dove there. In the Great Barrier Reef, since that video that you just saw, those exact same reefs have since then either bleached or died in what has been the most acute global bleaching event on record. And diving in the Red Sea wasn't so uplifting either. Actually, the sites where I was trained as a scuba diver were now closed to the public because of their degraded condition. My personal narrative reflects the situation for many coral scientists. These scientists must deal with the biopolarity even much more intensely, as some of them have to make decisions about which corals to save and which corals not to save. And they must make these decisions on a daily basis. When reading the barrage of depressing reports from the Great Barrier Reef, I wondered how, in the face of such devastation, these scientists find the motivation required to proceed in their dedicated work. What's the point in saving corals if they will eventually die of global warming and ocean acidification? Anyway, I remember asking Nilda Jimenez, who was my first interviewee in this context, and an employee of Puerto Rico's Department of Natural and Environmental Resources. Her weekly labor included tying corals together with dental floss to affix them to the ocean floor in an attempt to restore them from disease. Jimenez pointed out that the only other alternative was doing nothing. This was not something that she could live with, she told me. A couple of years later, marine biologist Nancy Knowlton of the Smithsonian Institution tweeted along the same lines. Bleaching is bad, but giving up is worse. This kind of proactive approach made sense to me. It was inspiring even. But just a few interviews later, a less optimistic scientist who defined himself as a realist, as they all do, swayed me to the other extreme. He told me passionately that instead of wasting time restoring coral fragments who will then die again from similar stressors, we should focus our attention on the real change that needs to happen, stopping emissions. Anything else won't really matter in the long run, he told me. And so I have been swaying, along with my interviewees, from hope to despair, from despair back to hope. This part of the swaying, like the oscillation, was difficult enough emotionally. What I didn't expect, however, was the extent to which the two approaches are alienated from one another. In a very necessary break between the sessions in Hawaii, one Australian, and I think it's not incidental that this is an Australian ecologist, he was wondering why I mentioned that he's Australian, and I actually will mention it again, he's Australian, complained about having so many sessions dedicating to what he referred to as the crap of restoration. Not a very nice word at all if you're not familiar with it. All right, so according to him, it is shameful that a professional conference would dedicate so many sessions to such an unscientific topic like restoration. So in the next 25 minutes or so, I would like to ask for your patience as we oscillate together with the scientists from hope to despair and then back to hope. So I will start with the narrative of hope, and we will focus on restoration. We will then move to the despair side of the pendulum and focus on bleaching and monitoring bleaching, and come back and discuss assisted evolution. This is kind of the most, I think, radical project of the genomic turn in coral science right now. So overall, I just want to say that I'm interested in the existential crisis that coral scientists are experiencing when they are put in the position of not just learning about and gaining knowledge about corals, but also speaking for their corals in order to save both the corals and themselves from extinction. So let's start with coral nurseries, the hope side of the pendulum. This is Sara Frias-Torres, and she grew up in Barcelona and spent her childhood on the Mediterranean. I learned how to snorkel before I learned how to swim, she told me in our interview. Here's how she explained why she transitioned from being a marine biologist working in a lab to becoming a conservationist and then to working in a nursery in Seychelles. And I'm quoting, "When I began scuba diving as a marine biologist, the Mediterranean that I was seeing wasn't the one that Jacques Cousteau showed on television. It changed completely. There were no fish. And so that's got me to say, okay, I need to work on conservation. But there comes a point when you see that you've got to do more. So now I'm both in conservation and in restoration." So just notice the distinction that the scientists make between conservation and restoration. And I want to speak to the tensions between these two fields that may not be readily apparent to us non-coral scientists, right? So just to explain briefly that while conservationists have traditionally been advocating preservation and protection of habitats and species with minimal human intervention, restoration ecologists advocate more active interventions by humans, what some also call gardening. Frias-Torres and I communicated extensively over email. We finally ran into each other at a festive dinner in the Hawaii meeting I spoke to you about before. The singing, dancing, and clapping, this was in the evening, it wasn't the entire conference, didn't stop Frias-Torres from outlining her detailed agenda for saving corals. So just think about Hawaiian dancing and a lot of Hawaiian music, and Sarah just yelling to me and me trying to record her and to document what she's saying over all this happenings, all right? The restoration of the planet, this is what she yells to me, the restoration of the planet is the greatest challenge of our century, of our lifetime. If we don't work on it, there is no future. So Sara Frias-Torres is a passionate believer in the power to restore, and her passion was contagious, but she also was very clearly frustrated. Many of her colleagues from the traditional coral conservation world give restoration folks a hard time. So here is kind of what I witnessed the night before this exchange, when there was like a little question and answer session during an evening panel. And so Sarah raised her hands and asked and kind of made the following statement, which I thought was very provocative. And so I'm quoting. And you can see it on this slide as well. "I learned this from a pediatrician working in poor villages in South America. He said that in the emergency room, they don't have the resources to attend to all the babies and little children at once. When in a triage situation, they only help the babies that are crying. Because if they're crying, it means that they're strong enough. They still have some life in them, that the medical treatment will be useful. For the babies that don't cry, there's no help. We are now in a triage. We can only save the coral reefs that are still crying for help, because they still hold enough life that the conservation effort will be useful. What to do with the others? As a coral reef restoration scientist, I ask you, give us the rejects. Give us the dead and the dying. We'll patch them up. Conservation and restoration should work together. In this way, coral reefs will still have a chance at survival." So very passionate. Strangely enough, after this statement, there was a very uncomfortable silence. And a few very long moments later, the next question was asked about a very different topic, and the conversation moved on. The plea to accept restoration as a legitimate sister rather than as an unwanted child of conservation was avoided this time. The tensions surfaced again shortly after when the facilitator asked the so-called doom and gloom expert, Ove Hugh Goldberg, whether, in light of the precarious state of corals, all strategies to save corals are now game. Hugh Goldberg responded with a graph. He argued that even if we balance out emissions, temperatures will still climb until mid-century. So even if we balance everything out now at 0.9 degrees Celsius, it will still climb to at least 1.5 degrees Celsius. So he's saying there's no sense to do restoration right now when the corals are still going to get fried, right? We need to first wait until that graph balances out and only then start doing restoration. At the moment, he emphasized, and I'm quoting, "It makes no sense because you put corals into the ocean and they grow for a while and then bleaching comes along and they die." A member of the audience fiercely objected to this statement. The right moment to do restoration was yesterday, he insisted. And so while this debate is still going on between conservation and restoration, between 2013 and 2015, Freya Storace and her team cultivated 40,000 corals in the African island nation of Seychelles in the Indian Ocean. So this is Freya Storace, and she's working in a rope nursery in Seychelles. She managed to transplant, after two freak events that killed 7,500 of her corals, she managed to transplant 25,000 into a degraded reef nearby. And according to Freya Storace, that reef increased in fish numbers and even new baby corals settled in that reef, which is kind of a sure sign for restoration. So let me take a moment to zoom out and speak a little bit more broadly about coral propagation and restoration, so to give you a context for this work. So the range of sites for coral propagation is very varied, and so do the reasons for this propagation. Corals are propagated for conservation purposes, for public education exhibits you may have seen in aquariums and in zoos, for profit in the ornamental trades. Some of you may have seen necklaces or jewelry. For research in biomedicine, which I will kind of give you two images of in a second, and for the private hobbyist industry, which my daughters are pushing me to become part of. As a tool in conservation management, cultivated corals can be transplanted back to the reef, usually into damaged areas, improving the recovery of corals suffering from degradation. This form of management is referred to as restoration, and some have suggested calling the specific two-step process of growing coral fragments and then transplanting them coral gardening. Massive underwater projects include using nursery cultivation and transplantation to restore reefs, as well as eliminating pest, and so I'm using these terms with quotation marks, pest and invasive species such as the crown-of-thorns starfish in Australia. So here's an image of the crown-of-thorns starfish, which sucks the life out of the coral reef, of the reef-building corals. And this is a very interesting story. I'm not going to tell it here, but just so you see the image. This is the Cotsbot, developed in Australia. This is a robot that is designed to kill the crown-of-thorns starfish. It injects the fish with the starfish with specific poison, that salt that kills it on the spot. So they've got permit to kill more massively than humans can do, and through storms, etc., whereas humans cannot necessarily dive in those conditions. I can speak more about it if you want later. So the crown-of-thorns starfish is actually not considered an invasive species. It's so-called a pest. It has these blooming episodes right now in the Great Barrier Reef. This one is considered by traditional conservationists as an invasive species. This is the lionfish, as you may recognize, and apparently very tasty. And actually, people are paid to kill it or cull it now in the Caribbean because of its massive kind of taking over of the ecosystem. Restoration sites range from locations selected for their ecological significance to designing artificial reefs for tourists. So here's an artificial reef that Ariel and I dove at. Do you remember that, Ariel? This is the Tamar Reef in the Red Sea. And then there's also these mini projects that hotels are now doing to kind of let their guests experience what it means to be an active conservationist. So guests come, they pay money, and they plant corals. And they can even engrave their name in a coral concrete frame so that their name will be there forever with a coral. If the coral survives, right? But at least the concrete frame will. So restoration projects vary not only in aim, geography, and target species, but also in techniques. Corals can be restored on metal. They can be restored on ropes, and you see this is from Eilat, and this is again from Seychelles. They can be restored on nets. I couldn't find a photo. And they could be restored on PVC trees. This is a really interesting example. Again, I can talk about it later. From the Florida nurseries, there's a person there, the director of the Coral Reef Foundation. His name is Ken Nedemeyer, and I actually use his narrative quite extensively in the book because he's a very interesting person, and how he came up with this idea of planting corals on trees. And this whole idea of the relationship between silviculture and kind of marine ecology is very interesting. So he plants them there because apparently they grow quicker, they get less infected by less disease, and actually the fish like it. So and it doesn't, and there's issues, legal issues too. So the turtles don't get entangled in it. So this is kind of why they came up with this strange structure of having like these tree nurseries underwater. All right, so whereas these nurseries in Florida and the one I visited in Culebra propagate the Acropora corals for restoration purposes, the Acropora are now considered threatened according to the Endangered Species Act, and so that's what they focus on. This one that you see in the image, this is the Cervicornis. It's kind of a branchy coral. And they also do the elkhorn, which is kind of, I think if you remember, we saw before, is kind of a more like, has a more massive cover. It's harder to propagate. So I will give you some images. This is from the farm in southern Israel, in the middle of this desert. Again, Ariel came with me to that field work. And it's really literally in the middle of, like, you won't believe, like there's no water sources or anything, but then there's a farmer, and what he cultivates is not peppers, but corals. And so this is him holding some of the corals, which he's using for the biomedical industry. Apparently corals are more like us, so they're really good for human bone and dental implants. And so he's raising them for implants. But the largest coral farms in the world are situated in Indonesia's ocean waters. So this is another one of the OK coral farm in Israel, and here's the image from Java, Indonesia, where corals are manufactured in large numbers for the coral trade. One can also find corals and their symbiotic algae at university and research labs. And this is only 5 minutes away from my office, and so whenever I get a desire to smell ocean, I go to the fridge in that lab and I open it up, and there is a strong smell of algae, because this is the largest Symbiodinium lab. Symbiodinium is the name of the particular algae that is collaborating with the coral animal, and I'll talk about that in a second. So this is a collection, the largest collection in the world of symbiotic coral algae for purposes of research. So, this was the hope side of the pendulum. Are you ready for some despair? Okay. As I mentioned, coral reefs are a symbiotic organism. Coral scientists refer to the coral as a holobiont. I don't know how many of you are familiar with this kind of very biological term, Lynn Margulis, all that kind of trajectory. Those of you who read chapter 5 of my book probably are, because I focus on it there. But a holobiont is basically a conglomerate of animal, algae, bacteria, and viruses, all part of this one organism who can photosynthesize with the help of the algae. So this is an animal that can do photosynthesis, right? But it can also use its tentacles to feed on the zooplankton or whatever other stuff that goes on in the water. So it can do both photosynthesis, as we're talking about reef building corals, and eat with its tentacles, and that's why it's called cnidarians. The meaning of the term is to sting. So while most cnidarian stony corals have very small polyps, the polyps average about 1 to 3 millimeters in diameter, the entire colonies can grow to be a few tons in weight. And here, for example, is the second, I couldn't find the first, but the second largest known brain coral in the world. And brain corals grow very slowly. As they grow, these reefs provide structural habitats for hundreds of thousands of different vertebrate and invertebrate species. Coral scientists say that although reef-building corals constitute 0.1% of the ocean floor, they provide 25% of the ocean's biodiversity, a quarter of the ocean's biodiversity. And this is why they are referred to as the rainforests of the ocean. So kind of a rainforesty picture. So what is bleaching? Bleaching is what happens when the relationship between the algae and the animal is interrupted. And so it's not clear who's the agent here, but the algae leaves the coral. It's not clear if the animal expels the algae or the algae decides to be expelled, but it's clear that the relationship is interrupted and that causes bleaching. This is a bleached coral. Okay, well, actually this shows you where the algae is situated within the tentacle. So it's in kind of the outer wall of the polyp. This is one coral polyp in a colony. And this is the process of bleaching. A healthy coral you see on the left with the algae on the sides. You see that it's colorful. Then the algae is expelled or expels itself. And the bleached coral is the one on the far right, the white coral. This is a bleached coral. Now, mind you, bleach does not mean dead. This is a kind of two-stage at least, right? I don't want to be too simplistic, but at least two-stage process. Bleaching could be reversed under certain conditions, but for the most part, bleaching means that the coral is on the path to death. And those are interesting moments, the process of dying, right? So it's not like alive and dead, but it's alive, bleached, and dead. So bleaching events are quite a modern phenomenon. So they've been recorded since the 1980s in kind of small episodes that are more local and regional. But the U.S. National Oceanic and Atmospheric Administration, I always have to grind my teeth to say that, and I'll call them NOAA in short from now on, right? So this administration has for some reason the power to announce global bleaching events, and I still haven't figured out what their criteria is for announcing these global bleaching events, but until now there have been 3 global bleaching events. The first one was very recently, 1998, and at that point 16% of coral reefs around the world bleached. The next one was triggered by the El Nino of 2010, and again it was massive, but nothing was as massive as the bleaching event in starting in October 2015, which has been going on, and I won't spoil what's going to happen. So, but at least 2 years, right? And I'm showing you a few images from the 2015 through 2017 event. So this is the bleached coral, quite a spectacular, image, and this is the dead coral, which just means that it's covered with algae at this point. So again, healthy, dying, dead. So the corals are very vulnerable to thermal stress, and we're talking about just a slight elevation of the degrees in the ocean, right? This is now we're only at 0.9 degrees Celsius. So what will happen when the temperature increases by more, that's still being kind of discussed and debated. The director of NOAA's Coral Reef Conservation Program was quoted saying, "We have boots on the ground and fins in the water to reduce local stressors. Local conservation buys us time, but it isn't enough. Globally, we need to better understand what actions we can all take to combat the effects of climate change." So this is a tension between the local and the global that a lot of coral scientists speak about. Is it enough to do local, or do we have to lobby the politicians to actually make a difference on the global scale? And here are a few slides from actually the media and how the media has interpreted the latest global bleaching event, which I found to be quite interesting. So these are just a few images from The Guardian, from the Washington Post. A catastrophe laid bare. More than 90% of the world's coral reefs will die by 2050. Large sections of Australia's Great Barrier Reef are now dead, scientists find. And this is the last one that caused a lot of concern among the scientist community, the obituary for the Great Barrier Reef that lived for 25 million years until 2016, right? So quite dramatic. So it was interesting to me that everything was it was predicted. The bleaching was predicted, and I'll tell you in a second how it was predicted. But although it was predicted so accurately, the level of bleaching caught scientists, especially at the Great Barrier Reef, caught them by surprise. It feels like a very special person whom you just started to get acquainted with suddenly dies on you, scientist Joni Klepas told me about the coral colonies in Costa Rica that she has been working with. I feel so helpless, she added. So last week, just June 19th, I believe, was a bad and good week for coral reef building corals. So on June 19th, NOAA declared that the global bleaching event is likely over. This was just last week. Oh. But the bad event was that UNESCO announced that almost all of the 29 coral reefs on the UN World Heritage List have been damaged by bleaching and are on the course toward death. So this actually happened 3 days ago. So like I said, let me say a few things about monitoring. And then we'll go back to hope. Okay, so bear with me with the despair. So Coral Reef Watch is an organization founded by NOAA, and it was created after the first global bleaching event in 1998, And using climate monitoring satellite data, Coral Reef Watch informs marine park management and scientists which corals may be at risk for bleaching. So they really collect data, and based on the data, they decided that that the corals are most susceptible to bleach when the temperature of the sea rises to exceeds the average of the typically warmest month by 1 degree Celsius. So if in the warmest month it's 30, then if it exceeds to 31 degrees Celsius, then the corals will bleach. So this is how they record or they predict the bleaching. And they took this data and did an online experimental chart displaying where these areas of high sea surface temperature will be found in real time across the tropics. So these are some of the maps that they produce through these calculations that declare the red is the hottest, right? The blue is the coldest, that declare, that predict what are going to be the hotspot charts in almost real time, right? And this is the one from last week, I believe. Since I subscribed to this free email alert, so from the 227 sites that NOAA monitors every 15 minutes, right, and then produces these charts, they send emails. So you can actually go for free and get an email that tells you, that gives you an alert if your reef is getting to the red zone. But at a certain point, I started noticing that I'm not opening the emails anymore because it was too devastating. So now I just glance at the titles and I just move on. And this reminds me of something that Nancy Knowlton, who is the mother of ocean optimism, I guess some of you know her, that's great. So Ocean Optimism received more than 59 million posts since its initiation in 2014. And this is what she told me in an interview. And I'm quoting, "You present people with large problems and no solutions, and they just go to the bar. Why get upset about something that you can't do anything about? There are lots of stories in scientific venues that even I don't bother to read. I know it's bad news, with just more details about how bad it is. There's only so much doom and gloom that I can carry." But her colleague, Ove Hugh Goldberg, disagrees. From his perspective, current conservation efforts are like reorganizing the chairs on the Titanic. What we need instead is to change the ship's deadly course, he told me. Not enough to rearrange our chairs. So what can scientists do in the face of such a spectacular and massive death? Terry Hughes, who led the efforts for documenting the recent bleaching events in the Great Barrier Reef, tweeted at the same time that he was kind of documenting from the helicopters. And this is the tweet that he sent out. "I showed the results of aerial surveys of bleaching on the Great Barrier Reef to my students, and then we wept." So I think this statement kind of captures the existential and very emotional crisis that coral scientists are currently experiencing on the despair side of the pendulum. This is the news item in the Washington Post that came out after Terry's post, and then we wept, right? And finally, to conclude the despair side, coral reefs are dying. Seacorps is a nonprofit organization that includes a lot of scientists, and this is kind of what they say is happening. So are you ready for hope? Let's go back to hope and we'll wrap up there. Assisted evolution. In the final leg of my trip to Hawaii, I was invited by the president of the International Society for Reef Studies and the director of Hawaii's Institute of Marine Biology, Ruth Gates, to spend the day with her on Coconut Island. Again, such a difficult time, right? So this is Coconut Island off the shore of Oahu, and this is inside the lab. Although she first studied and then worked with Ove Hugh Goldberg, who's the doom and gloom guy from before, Mr. Doom and Gloom as he's called, so she worked with him for many years, but her perspective on the situation is very different. From her perspective, and I'm quoting, "We will either have a reef in the future or we won't. But if we continue down this trajectory and we do absolutely nothing to assist the system, it's likely that we'll have nothing." Close quote. By assist the system, Gates is referring to a radical proposal that she developed together with Madeleine Van Oppen to captive breed corals that have survived the bleaching and who are more tolerant to climate change, or specifically to heat. These are the supercorals of the future, as the two women call them. I thought about designing a supercoral, but it wasn't enough time. Maybe for the book cover, all right. And the process of propagating and introducing them into strategic locations in the reef will accelerate, as Ruth Gates says, natural processes that may currently be too slow to deal with the rapid pace of human-induced climate change. So here from Ruth Gates, the basis of our project is that we are assisting nature. We are accelerating natural processes. We're not creating anything new. We're doing what nature does and just trying to find ways to do it more quickly. I'm interested in who, and she means which coral, is performing well now. If we take the ones performing well now, can we lift their biology to perform even better? Can the best performers now become the best performers in the future? We're doing that through selective breeding, just classic selective breeding. It's very interesting how she really wants to highlight that, and we can talk about that later. We're interacting very hardy genotypes, and then we're going through conditioning, running our corals on environmental treadmills with the idea that what doesn't kill you makes you stronger. Gates is expressly collaborative and supportive of different attempts to save reefs, including restoration. But she's critical of the doom and gloom side of the pendulum. This is what she told me about a conversation she had with one of the scientists that I mentioned before, and I'm not going to mention names here. As I said to him, "I hate to be blunt, but you think that the restoration and assisted evolution need your blessing, and you know what? The reality is that you are so behind the fastball. Restoration is happening. Assisted evolution is happening. We are doing it, and guess what? If we don't, there are places that will have no reefs left now. Christmas Island has no goddamn corals left. Are they going to wait for you to give them permission before they start doing these things? Absolutely not. Gates does have one major criticism toward the restoration community for focusing too much or too little on coral genetics and epigenetics and focusing too much on asexual reproduction. So what they do is they just fragment specific genotypes of corals and they don't really do much in terms of genetics. And from her perspective, that is the wrong way to go. She's a geneticist and she believes that genetics are the hope and the future for corals. She just got $4 million from Microsoft co-founder Paul Allen, and she and Van Oppen have been designing experiments to condition corals to stressful environments with the aim of inducing epigenetically controlled stress tolerance that would be passed on to the next generation. Both went out of their way to stress that this is not genetic modification, this is not a GMO. Rather than tinkering with the genes, they are merely doing what humans have been doing with wheat and tomatoes for centuries, crossbreeding for selected traits. We have to be realistic about where we are, Gates told me. For the most part, coral reefs are almost all declining. That means it's not sufficient to step back and wait. People tell me that nature might do that by itself. It might. But it doesn't seem to be, not quickly enough. So I would like to show you a quick video of hers. Turn it down.
This is all the selective breeding setup.
Yeah, I may speak over her for the first 40 seconds because we're chatting. Right. So this is in Coconut Island, right? In her lab, and you will see she is very nice to everybody, and so everybody that comes in is a new world. We will start in a second with a more serious part, right? Okay, here we go. Can you hear?
Because we're going into a spawning next week, everybody's cleaning up, ready for the big system. Behind you, kindly, is all the controls for all of the simulated ocean conditions. These are all the valves that control the environment. And we're essentially controlling, as you know, CO2, the level of acidification and temperature. And we can monitor our tanks remotely from the inside here to make sure everything's going on that we want it to out there. And we have sensors out there that tell us if anything goes wrong in here. So this is like an engineering feat, quite frankly. And the outdoors, we're going to have a look at tanks outdoors in a second, but inside here, we're retrofitting this entire tank setup now. It was working a long time ago, and then we are now refitting it for working with much smaller animals. So you can see the tanks are much smaller. What we're really trying to achieve is adults, quite large fragments outside, working with much smaller individuals inside, exactly replicated conditions.
Smaller coral individuals.
Smaller corals and thin juveniles that we're growing. Obviously, our selective breeding over here.
So, you'll tell me a little bit more how these machines, still technologies work.
This is really just keeping clean. So they basically swim around. You can basically, there's a flow that goes through that keeps them in one side. You can lift that, you can clean them and put them back in. There's nothing in there right now.
So it's going to be with water and
it's full of water when it's all
and you have to tell me the others.
Water.
I think
All piping in.
this is just from the bay.
No, this is all from our simulated conditions, so we can maintain the simulated conditions in the selective breeding process. Hello?
Okay, this is where other people come in. So a day with Ruth Gates meant an overdose in positive thinking. She picked me up from the pier for our day's tour and walked me around the island. Everyone we came across was delighted to see her. She knew the life stories of everybody she came across, from the garbage collector to every volunteer on the island. She greeted them warmly and knew their life stories inside out. And this is what she told me. People want to do something, they want to help. The gloom and doom is paralyzing. The scope of the climate change issue is so paralyzing. But not so for Gates, interestingly. As befits her black belt in karate, she has taken on the challenge of climate change full force. And as with karate, she tells me, most of the battle to save corals is mental, not physical. I want to start a movement, she revealed in one of our many animated conversations. There's people who do that for a living, who manage campaigns. I don't know how to do that, but that's what saving corals would entail. We need to empower people to do it. So I would like to end my presentation with these words, and thank you for your attention.